Welcome to this explainer, where we're jumping right into the incredible cellular choreography of meiosis. Have you ever looked at a family photo and thought, how on earth do I look so completely different from my siblings when we share the exact same parents? Well, the secret is buried deep inside your cells, in this wonderfully complex, highly specialized process that literally rewrites the genetic script every single time it fires up. Today, we are going to break down exactly how it pulls off this magic trick. Here's our roadmap for today. We'll kick things off with the actual purpose of meiosis, get our key vocabulary sorted, and then take a really deep dive into prophase 1. After that, we'll see how meiosis 1 and 2 finish the job, and finally, contrast this whole thing with mitosis. All right, section 1, the purpose of meiosis. Why do cells do this? Before we get into the heavy microscopic details, we have to talk about the big why. Look, meiosis is fundamentally the engine of survival and diversity. Its main job is to produce haploid gametes, so your sperm and ovum. By slashing the chromosome number right down the middle, from our normal diploid count to a haploid half count, meiosis ensures that when two gametes eventually fuse, the resulting zygote has the exact right, stable number of chromosomes. Because think about it, if our cells didn't do this, our chromosome count would just double every single generation. But it's not just a biological math trick. It uses things like independent assortment in metaphase 1 and a wild process called crossing over to make sure absolutely every gamete is entirely unique. Okay, moving to section 2, key meiosis vocabulary. Let's meet the cast. To really wrap our heads around the action, we need to know who the players are. What we're looking at here is a single chromosome that's duplicated itself into two identical halves. We call these identical halves sister chromatids. Notice how they're basically hugging? They are bound super tightly together right in the middle at that pinched area called the centromere. As long as they're joined up like this, we consider them one single duplicated chromosome. Next up, the homologous chromosome. You know that we inherit one chromosome from our dad and one from our mom to make a pair, right? Well, these pairs are homologous. That means they are a perfect match in their overall size, their length, exactly where that centromere sits, and the actual types of genetic traits they code for. So when you look at a homologous pair side by side, it's actually two separate duplicated chromosomes hanging out together. The maternal and paternal chromosomes each bring their own set of those identical sister chromatids we just talked about. Now, things get interesting when these homologous pairs actively seek each other out inside the nucleus and physically link up. That pairing up process, it's called synapsis. And once they're officially a couple, which means we've got four chromatids together in one spot, biologists call that structure a bivalent or sometimes a tetrad. Just look at how flawlessly the maternal and paternal chromosomes align during the synapsis phase. They are literally matching themselves up, gene for gene, getting ready for the absolute coolest part of genetic reproduction. Because once they're locked in this tight embrace, crossing over kicks off. This is a physical, literal swapping of genetic material. Non-sister chromatids, so one from the mom side and one from the dad side, actually break and trade pieces. And the specific, visible, cross-shaped intersection where they make this trade is called a chiasma. You can see the whole sequence playing out here. They overlap, they trade at the chiasma, and boom, you get completely unique recombinant daughter chromosomes. A little piece of the maternal chromosome is now permanently stitched onto the paternal one, and vice versa. This is exactly why you and your siblings are different. Your parents' bodies are actively shuffling the genetic deck before dealing the cards. Which brings us to section 3. Deep dive into prophase I, the main event. Now that we know the lingo, let's unpack the longest, most critical phase of the whole process. Prophase I is honestly so action-packed that biologists divide it into 5 distinct substages: leptotene, zygotene, pachytene, diplotene, and diakinesis. Each one triggers a very specific sequence of events, so let's walk through them. We kick off with leptotene. This is when our replicated chromosomes finally start to condense and reveal themselves under the microscope. They look like these really long, incredibly fine threads winding all through the nucleus. They haven't paired up with their homologous partners yet. Right now, they're just packing themselves tightly. Then we move into zygotene. This is where synapsis officially begins. A specialized zipper-like protein structure, called the synaptonemal complex, starts forming. Think of it as biological Velcro that physically grabs those homologous chromosomes and zips them tightly together along their whole length to form our bivalents. Once that zipper is fully closed, we are in pachytene. Guys, this is it. This is the exact moment crossing over happens. The non-sister chromatids are actively exchanging their DNA. This stage is absolutely crucial. Without Pachytene, all that incredible genetic variation that drives human diversity just wouldn't happen. As we wrap up prophase I with diplotin and then slide right into diakinesis, that synaptonemal zipper starts to break down. The homologous chromosomes try to pull apart a bit, but they can't quite escape each other because they're still firmly pinned together at the chiasma, those physical crossover points marking exactly where the DMA trades just went down. Okay, so we've successfully shuffled the deck and built all this amazing genetic diversity, but that leaves us with a massive mechanical problem. How on earth does the cell physically divide up all this tangled recombined material to actually make functioning gametes? Let's find out in Section 4, completing meiosis 1 and 2, the cascade. To solve the tangled chromosome problem, the cell fires off two rapid back-to-back rounds of division. First up is meiosis I, which is laser-focused on separating those homologous chromosomes we just spent all that time pairing up. It literally drags the newly recombined maternal and paternal chromosomes to opposite sides of the cell. Then it dives straight into meiosis 2. And here's the kicker. It divides a second time without bothering to replicate its DNA again. This second round acts a lot more like standard cell division, where it just rips those sister chromatids apart. The end result of this whole cascade? 4 completely unique haploid daughter cells. And finally, Section 5, mitosis versus meiosis. The showdown. To make sure we've totally nailed this, let's stack meiosis up against mitosis, which is the everyday cell division that heals a scraped knee. Mitosis happens in your somatic cells, executing just one division to pop out 2 perfectly identical diploid clones. Meiosis is totally different. It's exclusively for germ cells. It forces 2 complete divisions, and because of that wild synapsis and crossing over back in prophase 1, it generates 4 haploid cells that are genetically distinct from each other and completely different from the parent cell. Which brings us to a pretty mind-blowing thought to leave you with today. Because meiosis relies on the completely random, independent assortment of chromosomes, plus the physical, chaotic swapping of DNA during crossing over, the number of possible outcomes is basically infinite. When you crunch the math on those odds, the exact genetic blueprint that makes up you has literally never existed before in the history of the universe. And it will never, ever exist again. Pretty wild to think about, right? Thank you so much for hanging out with me for this explainer. I really hope this gives you a whole new appreciation for the microscopic wonders happening inside of you right now.